So I'm going to title this video something slightly different from the initial question, but I'll explain what I mean as I do that. And that is, nope. Conducting versus respiratory divisions. And of course, that is conducting versus respiratory divisions of the respiratory system. So the initial question is essentially talking about alveoli versus bronchioles, so two very little tiny structures in the lungs, and whether or not their function is the same thing. And this is a nice tie-in to what you're going to be looking for in the histology slides. I'm not going to refer to those histology slides for this video, but I am going to draw out for you what kinds of cells are in those locations, because that's really what's going to determine whether or not that part of the respiratory system performs gas exchange or is just carrying air to a place where gas exchange can take place. So to start out, let's actually just define these two things, and then I'll start to do some drawing. So conducting division carries air in bulk flow, and I'll explain what I mean by that in a minute. So carries air in bulk flow, what I mean when I say that is the same way as like air would move through the duct system in a building. So it's moving in a mass through an enclosed tube-like space. Respiratory division is where carbon dioxide and oxygen are exchanged. And this happens across cell membranes. So even though carrying air sounds like it would be the same thing as gas exchange, they're not the same thing. So this is our definition of conducting versus respiratory. And now I will start to show you in drawing what I mean. So we're going to draw some different tubes and whatnot. So let me draw just a really basic picture of a trachea and two bronchi. And we got some lungs there too. Okay. So when you breathe in, you're drawing your diaphragm down and air is entering. And what that does is sucks atmospheric air into your airway. And the atmospheric air is just a mixture of a bunch of different kinds of gases. So it's mostly nitrogen, and then there's a little oxygen and a little less carbon dioxide and some other trace gases, as well as water vapor. So not all of the air that you breathe in is air that's useful to you. It's only a small fraction of the air that you breathe in that's useful to you. So trachea and bronchi and most bronchioles are part of the conducting division. And that's because, again, they're just having air move through them. So no molecular gas exchange is taking place there. So what I'm going to do is draw some very basic histology. So we're going to start with a drawing of a bronchiole. And I'm going to draw the little cells that line the bronchiole as well. I'm going to be a little bit lazy about my cell drawing because I don't want it to take super long time. But as you can see by the way I've drawn the apical surface of the cells, remember, friendly reminder about histological terminology, because it's important to keep refreshing that. Apical surface means in an epithelium, the side of the cell that faces the lumen, not the side of the cell that faces away. So because I've drawn it all squiggly, that's me trying to tell you that this is a ciliated epithelium. And these cells, I'm drawing a really small bronchiole here. As you make your way through the airway from larger air tubes like the trachea towards smaller air tubes like terminal bronchioles, for example, the histology of the epithelium goes from being pseudostratified ciliated columnar epithelium with lots of goblet cells, and then it sort of gradually transitions as the tubes get smaller to a ciliated columnar epithelium, just regular columnar, and then eventually right before it switches to the respiratory division, it does appear cuboidal. So this is something that students don't often expect, is there to be sort of a gradual shift in the histology of something. There's no clear boundary between one area and the next. You just see this gradual change in the epithelium. But regardless, you need these columnar cells with the cilia because they're part of the way that your lungs and airway clear themselves of debris. So you breathe in particulate matter all the time. Whether or not you live in a filtered air environment, you're still breathing in dust and stuff. And that dust gets caught in the mucus that is produced by your respiratory epithelium. And then that mucus with trapped particles is basically moved back out of your airway by the cilia. So it's a really important part of the histology of the airway that serves a very specific purpose. Alrighty, so then outside of the bronchiole, so I'm going to label this too. We've got smooth muscle. And that's going to be basically responsible for changing the diameter or the distance across this tube. And there's no cartilage here because this is a bronchiole, not a bronchus. So as we continue, I'm going to follow one of these trajectories. I'm not going to draw the entire thing. So what you end up seeing is this gradual sort of decrease in height of the epithelial cells, and they lose their cilia and they start to look more cuboidal. So the morphology of the epithelium changes. We still got smooth muscle. And now we start to see alveoli popping up. Actually, I want to use that for drawing. So just all of those, I'm only indicating 2, but they're all alveoli. So alveoli are just very thin little balls. I'm drawing the cells that make them up. So there's squamous epithelial cells. Squamous meaning scale-like or thin and flat. And then of course on the outside of these, really, really close, they're separated only by a basement membrane. We've got blood vessels. And these are going to be branches. Actually, I'll ask you, for those of you who entered when I was explaining, I am recording this. So if you want to answer this question, do it in the chat. I'm just drawing these blood vessels. So I'm going to put branch of blank for the red guy and branch of blank for the blue guys. Anybody have any idea what those branches will be? Yes, vein, but more specific than that. They're branches of some veins and arteries from the cardiovascular system chapters that you should be familiar with, and I'm trying to help you connect those two. Blue is indeed low oxygen, but what vessel coming from the heart carries low oxygen? Pulmonary artery or vein? Pulmonary artery, very good, yes. So remember in the pulmonary division, the colors are switched because blood is going away from the heart, but it's oxygen poor. And so yeah, the blue guys here are gonna be a branch of the and the red ones will be branch of the pulmonary vein. So these are just the little branchlets of vessels you're already familiar with. So if you think about what I've described the function of those vessels being before, we know that pulmonary arteries are carrying oxygen poor blood to the lungs to get oxygen, and pulmonary veins are carrying oxygen-rich blood back from the lungs, carrying oxygenated blood to the heart to be distributed. The place where that exchange happens is at the alveolus. So let's draw, I'm going to draw some oxygen molecules and carbon dioxide. I'll make a key for myself over here. So the oxygen here is going from the airway into the blood vessels. And the carbon dioxide is being dumped from the blood vessels into the air. So I realize it looks a little bit messy, but what you should be seeing is basically the fact that oxygen is going from the airway into the blood, and carbon dioxide is going the opposite direction. I'll talk about the macrophages in a second because they're not central to the function of alveoli. They're very good to have, but they don't have anything to do with gas exchange, which is the main alveolar function.
Can you say again oxygen is going here, CO2 is going here. I was drawing, and so I'd like to write those words down.
Yeah, absolutely. So the arrows I drew are oxygen from the airway going into the blood vessels of the alveolus. So they're exiting air and they're entering fluid. And then the carbon dioxide molecules are exiting the blood and entering the airway. So basically, the arrows in the alveolus drawing here, that is trading of gas molecules taking place. You're taking useful ones from the air you breathed in and putting them in your blood, and you're taking, I don't want to say garbage ones, waste air, and moving that from your blood into your airway so you can then breathe it out.
I hate saying this out loud, but I hadn't really been thinking about how there would be blood on the outside of the alveoli. I think it's hard for me to integrate the different systems, and I feel really silly saying that, but it was helpful to hear you.
I would agree that probably most students don't make that connection on their own. So I think if you're having trouble with that, probably other people are too. So that's why this kind of stuff is important. So yeah, the whole, like, if you think about it, let's zoom out for a moment because you make a good point. Why would you have lungs at all if not to exchange with the environment? Because of course, you know, breathing in and out is great, but if there's no endpoint for the gas, you would just be wasting calories moving your diaphragm and there would be no benefit for it. So think of your lungs. They're just a way for you to harvest useful gas molecules out of the air. That's all they are. So you move the air into your inner lung surface using the conducting division, which is just a series of hallways for gas to march down. And then the respiratory division, which consists mostly of alveoli, and also these weird bronchioles that are kind of not completely alveoli. They have some regular wall. These are called respiratory bronchioles. So these two guys right here make up the respiratory division. Because respiratory division is any place where exchange of oxygen and carbon dioxide can take place. So you'll notice in the bronchiole over here, there is no arrow stuff happening because that's not true. One of the reasons for that is, of course, because the diffusion distance would be way too large. So remember from Biology 160, you guys did a little bit of this thing called the Fick equation, which was a little nasty bit of math that describes what determines the speed of diffusion. And one of the things that's in that equation is distance, meaning how far does a molecule have to go to diffuse? The bigger the diffusion distance, the slower the diffusion. So if you look at squamous epithelium over here, where the cells are really thin and flat, and the only thing separating these squames from the blood vessels is a basement membrane, that diffusion distance is small enough for you to exchange gas. Over here, we've got a big, tall columnar cell, and then a lamina propria, and then smooth muscle. Distance is way too big to exchange gas. So bronchioles and bronchi and the trachea, the conducting division, that's a hallway that air goes down. The alveoli are where the actual chemical gas exchange happens. So you could have lungs with bronchi and bronchioles only, and you would not survive because you would be unable to do gas exchange. So you need both. And then before I finish this, if you want to mix business with pleasure, I suppose I would say, I watched an episode of 2020 last night as I was falling asleep about this surgeon guy, and he basically faked he claimed he was able to successfully replace the trachea with an artificial trachea. And I was immediately like, something about this stinks. He was like, yeah, it's made of plastic. It's just, you know, like same plastic that water bottles are made out of. And then we go ahead and just install it where your old trachea was and it's good to go. And my immediate thought was, well, but what about the inside? The lining of the trachea has a very specific job with the cilia and the goblet cells and the mucus. And I don't see how he would replace that function. And it turns out that he didn't. And it was a huge fraud, and a bunch of his patients passed away just because they didn't have pseudostratified ciliated columnar epithelium lining their trachea. Which, you know, if I was overseeing that surgeon, that would have been like my first question, but maybe that's just me. But yeah, so that directly relates to this conducting division stuff, because although gas exchange happens here and anywhere where there's alveoli, the histology of the conducting division is just as important for lung health because it provides that mucus clearance function. So if anybody is here to become, for example, a respiratory therapist, these are the kinds of things that you're going to be thinking about and worrying about when you're interacting with your patients. So all of it matters. It's just that bronchioles and alveoli have different functions. So that's kind of the end of that.